All right, thanks everyone. Thanks for this great meeting. It's a very exciting time to be in neuroscience. So, arguably, one of the holy grails of neuroscience is to decrypt the neural code. And this is nicely captured by this, I'm sure a lot of you have seen it, by this quote from Francis Crick's book, An Astonishing Hypothesis, that says that something that we all believe in neuroscience, but still an astonishing hypothesis in the sense that all our feelings, our thoughts, what we do, what we see, it's because of something happening in the brain, right? The nerve cells firing, molecules, and so on. So in some ways, what we're trying to understand is how is the outside world and how we act. What are the correspondences between what's happening inside the brain and what's happening outside the brain? And now we are actually in a very interesting time because we also have other intelligent systems that are not biological, that are also in some ways behaving intelligently with respect to the outside world, whether that's the internet or it's an autonomous car driving around. You know, it's an intelligent agent that is not built out of biological stuff, but it's also interacting. And despite this progress, they still lack some fundamental qualitative differences that makes us think that there is something, there's some like source that is different in brains and artificial intelligence. And there's like a million demonstrations of this, but this is one of the ones that I like. If you take these dog and puppies and you change their arrangement, or you make a new collage that is not the standard on the internet, and you show it to the frontier models, they all pretty much fail. They either get it a chance or they get confused in parsing out these things. So you wonder, how is it possible that something that is like so smart, it can solve differential, any equation much better than most humans, it's incredibly capable of like segmenting complicated video. Tony's Ryoo can write really well these days. And then, it can still not solve this kind of problem. And, if you talk to people, if you look at the history of the field, the key is generalization. And I think this was brought earlier. And the point of generalization, this is one of my favorite reviews by Matthias Bethke and others in the collaboration where these neural networks are sort of, they get the generalization wrong. They lack certain amount of generalization. For example, the classical adversarial attack, where of this one here, if you put a cow on the beach, it gets it wrong. Or they have too much generalization. Like you can create these betterments that are not such betterments, but like this thing, it can make it look like another factor. And this is also, basically Johnny Depp tells his kid here that don't worry about it. We're just gonna like train it so much, download so much data, have so many people labeling it around that this won't be a problem. And this is really what the big companies are doing. And in some ways they're succeeding. And there's an argument to be made that they may be right? Maybe you don't need to really understand how the brain does it and all this stuff that we neuroscientists care about. You just download more data, get more people to annotate data, and you basically solve autonomous driving. And in some ways, I have, you can see the autonomous car driving around, and they do a pretty good job. However, it's an interesting time because these artificial agents are also built out of neural networks. They have components that sort of look like the brain. So what is it different? Like from a neuroscience point of view, we still want to understand what makes brains much more capable at solving these problems, or are they more capable? What is the difference? And it could be we can study the brain, and this is a whole field of neuroscience, has many levels of analysis. Like it has the substrate that is built is very different. It has all these beautiful cell types, it has different molecular components, the non-linearities are very different, the complexity of individual neuron, you can spend, write books and books on the differences, right? And their connectivity is much more complex, they have this stereotypical connectivity, but at the same time, there's some evidence that there's universal principles that are substrate independent. Like, for example, we know that you can build intelligent systems without relying on biological You don't need to build them out of neurons, right? You can just build them in silico. So that's one difference. But also, if you look at the inside of these networks, there is some evidence that, for example, this is a CNN, and you look at these sort of feature detectors, and these are macaque neurons in V1. You start seeing there is some similarities between them, despite that they're like, these ones you can just try any architecture you want, they'll still give you in these early layers something that looks like this. And if you look at the brain, you get this type of thing. Okay. So there is something that indicates that these representational level, where you abstract away all the details, you don't care about these molecules, ion channels, GPU, silicon, you don't care about the architecture, as much. You just like say, I want to trade systems that are capable of doing intelligent behavior, and I want to compare them at this representational level, okay, with mine. I think it's a very, very powerful level. And I personally think this is the level that in the next 10, maybe more years, where the more fruitful interactions between neuroscience and AI will be. Although I do work in my lab at the other levels, and I'll show you data again about that. I really think this is where the mind is. I think the other levels are, very interesting from a biomedical point of view, because that's where we have tools to manipulate the system. But this is the level where we can make glimpse to artificial intelligence. And this level, it started going back to Lord Adrian 100 years ago almost. Now I discovered the action potential and covered basically the representation of force in the outside world, how it enters the body, basically at the strange research level. And then you guys all know, the work of hardlining, Hee-Woon and Weasel, all the way to the Mosers. Many of these people got Nobel Prizes studying the brain at the representational level, in a way, finding correspondences between the outside world and what's happening inside. However, if you read the history of these people, going all the way to Adrian, they were all serendipitous discoveries. They were accidental discoveries. In fact, Adrian thought what he was doing was an artifact. He was giving basic action potentials. And he thought this is just an artifact. He was trying to debug and get rid of the noise. And then he suddenly realized the amount of force that was exerting on the muscle was correlated with the number of spikes, and he discovered literally that within 2 hours trying to debug the system, the rate code, and the action potential of this stuff. If you talk to Hee-Woon and Weasel, and I'll show you a movie, this was an accidental discovery, and the Mosers the same, and John O'Keefe was actually not didn't care about places. He was trying to study sleeping, he was getting frustrated that the mice were not sleeping, and as we were moving around, he saw the places. And this is a nice movie in 60 minutes. You can find it on YouTube, where David Hubel describes how they found this accidentally. They were using spots of light because they were students of hardline and Kufler, and they were using light, spots of light to study the retina. And then one day this light was dirty, and they found the edge detector. So now the reason is very simple. It's related to the previous talk and Blake, is that the behavior of what is possibly the input, let's say. Let's just look at the visual system. It's very big. You have just a monitor that is 256 by 256 pixels. You can generate an enormous number of patterns that you can, it's more than the number of electrons in the whole universe. So it's very hard to study systematically the brain, okay? And that's why if you look at these major discoveries, of course, hindsight is 2020. People have strong intuition sometimes, but generally speaking, they're accidental discoveries. And this is frustrating, because if you go back to the figures that I showed you, if you now take into account behavior, there's many modalities, the internal cognitive states, it just becomes this space infinitely large to probe it and understanding over one part of the whole brain is doing, okay? And this is a big problem. Okay. So, however, we are very lucky that in the last 10 years, there's been two parallel revolutions, not necessarily interacting, that I think could get us out of this potentially. I think this is some glimpse of hope. One is the Brain Initiative and other institutions have invested 10 years to develop neurotechnology. Like when I was a graduate student, there was very few techniques that we could use to scale up recordings. Now we have, for example, the light beam microscope from Ali Pasha Vaziri's lab at Rockefeller that we just replicated at Stanford, that we can record close to 1 million neurons simultaneously in an awake behaving mouse. And we have this wonderful technology that's built by IMED, NeuroPixels, that enable us to scale up and take it all the way to humans, okay? And we can scale up data collection in a way that is really unprecedented. But we do not know what to do with this data, how to just collect data, but we don't know how to use this data to make discoveries. But now at least we have these basically neural networks or foundation models or whatever you want to call them, that they enable us to take all this data and start building predictive models or one way or another, okay? Because if you think about it, what is neural networks, what is AI right now is taking human behavioral data, whether that's people writing text or driving around, and distilling that information or copying it on a chip via neural network to basically make predictions. You know, when you drive, you have to turn less than rate, when you label pictures, when you do next word prediction. It's all about predicting something from one data modality to another within a data modality. So we can just take all this data and ingest them into a model and start learning and making predictions. And in the last few years, and I'm just going to present work from our lab, and others have done similar work, is we developed this method that we call digital twins and inception loops to try and develop a new paradigm for neuroscience that we can sort of go away from the serendipitous discoveries and rely more on a systematic approach to basically decipher the neural code, okay? And I will just very briefly show this. This was the first paper that we published done by Edgar Walker, Fabian Sid, who is here in the room, and also Talia Muhammad, that basically this inception loop, this version of these experiments has 4 steps. One is you use technology to record large scale of data. For example, in these papers, we did 5,000 neurons with 5,000 unique natural images. So in a single experiment twice, you can get 25 million training points, let's say from specific brain region, using very deep pyramidal nodes. The second step is you use neural networks to just do the predictive model. As Blake said, we don't care about this architecture. We just want the best architecture. We're not trying to replicate it the way the brain is doing it. We just want to like get the best architecture. It's like system identification. We just want to get the best architecture to make the best predictive model. We don't care about how biologically, you know, how it relates to the actual biological mechanism, right? It should be in theory better, but we haven't, you know, there's a lot of reasons why it may not be right. Maybe in the future, okay? So once you build this, then you can ask the question, if you think about it, this is like what AlphaFold did, right? They use data, it was crystallography data, and they do the predictive model, but that was already very useful there because people cared about, let's say, mutation in an amino acid sequence, how to change the structure, and already that model by itself was used. But in neuroscience, it's not really clear to me at least, even if we get to this point, why is this useful? You know, we just copied the brain, right? Basically in a fancy engineering way. What are we going to do with it, right? So what is very interesting and I think very exciting is that despite this sort of concept that these neural networks are just black box, they're not just black box. There's stuff you can do with it to try and gain insights of how this thing works. What I'm going to spend the rest of the talk is like getting to some early insights that we've been using this type of models to try and understand some biology about the brain, and Catherine that's going to follow will give you a different set of experiments, and most of these are published or under or bioarchive. So the first thing that you can do, and I think you know Eva mentioned this, is you can just run a gazillion experiments in silico, right? Instead of now, if you could have a very good model of the brain, instead of like having, let's say, one graduate student running experiments for 4 or 5 years, you can have 100 million, you know, graduate students, and if this is in silico, you can scale it up, and what would take, I don't know, 1 billion years, you can run it in a few hours, and you have enough money and enough GPU. So that's one. The other thing is you can do experiments that are almost impossible to do in the brain. For example, because these models are differentiable, you can backpropagate the gradients through and do the Hee-Woon and Weasel experiment, or all basically a lot of neuroscience experiments, trying to find stimuli that modulate the responses of neurons in a systematic way. For example, one is trying to find the optimal stimuli. That's what Hee-Woon and Weasel were trying to do, right? And we're sitting there, we're just playing with stimuli, and they're trying to get the neuron to fire, right? That's what the Mosers did, right? They were like there in terminal cortex. I remember when I was a graduate student in Madhusson, I was in Madhusson, but I had friends there. They were studying the terminal cortex, and they had small boxes because space is more valuable at MIT, I guess, than in Norway. And they put it together already. It was like, in Norway, they have no space. Don't carry, they put a bigger box. How do you get a bricks, right? So how do you do this in a systematic way, right? So the thing is that what we did is, because the model is differentiable and you can backpropagate the gradient through, you can optimize the stimulus. And the first time we did it, we got these most exciting images that didn't look like abort filters. And we thought, okay, this makes sense. These models are hallucinating. They are sort of creating stimuli that are not exactly part of their training because we use full field images. They sort of zoom into one part of the field. So they're creating images that are sort of arbitrary. But then, we can run closed loop experiments that we call it Inception loop. You can synthesize the stimuli, show them back to the animal, and say, are the stimuli driving more than anything else I've tried? And got a lot of controls that you can read about it. And it did when you show the stimuli that look very exotic, but you can find them in natural images as patches of natural images. You get them to drive these neurons much stronger than anything we've seen, and they're very similar. The other thing that we've done recently, these papers on bioarchive, we looked at the invariances. This is work done. Unfortunately, I don't have the reference here. But we found these like neurons in mouse V1 that we think are important for segmenting information. They have this low high frequency sort of boundary between them. And they also have very interesting invariances where one part, half of it is like a simple cell, the other one is like a complex cell. And interestingly enough, something similar has been found by Chrysola from Anthropic and artificial neural networks. They call them low high frequency. So in the last 2 years, we thought, and like many others, is like, okay, instead of just training one model at a time, why don't we start combining data from many experiments? Because there are different ways to do it, but you can do it. In our case, it was all mouse visual text, many visual areas, many layers, many mice. And Eric Wang here built this, and he used different architectures. This is a convolutional visual transformer architecture with some stuff that takes into account the behavior of the animal. This was just a mouse running on a treadmill, the pupil, the eye movements. And basically what it's doing at the end of the day, for every single neuron across many, many animals, you have this gigantic network that they share a lot of features across layers, across areas, and then it just becomes a very good model of predicting visual responses. And we wanted to see how good is this model first. And in 2021 with Fabio and C, we had a similar paper combining data from many mice in a static version of this, and this is a more of a dynamic version. And one of these things that was interesting to us, and I think someone asked this question, is how well would this generalize to new neurons of mice? So what you see here is that you train it on these cohort animals. Let's say you have 4 animals, you train it with 150,000 neurons or so. And now you get a new animal, but you do not retrain the network again. You only just train the linear readout. But this is very important and very useful because if you want to do closed loop experiments, or we want to use it like that, it's very fast to train the linear readout versus retrain the whole network. Okay? And what we find is that on a single experiment on a new mouse, instead of taking 76 minutes to reach some level of performance, only with 16 minutes you would get the same level of performance if you use a pre-trained network. Now, to me, this is not that surprising because it's not like every mouse is different and every V1 is different. There's a lot of similarity, especially in the mouse. In the monkey, it's a little bit different, but in the mouse, you can look at these higher visual areas. We have a lot of similarity between V1. So the fact that it generalizes so well, to me at least, it's very exciting, but also it sort of agrees with our understanding of the functional organization of the mouse. The thing that was more surprising to us was that, and this is related to this idea, okay, if I train on one task, how much does it generalize to others? This is not tasks, these are stimuli. But if you train it with natural movies, it generalized to a bunch of other stimuli that were not used for training. In fact, some of them we did post, but we didn't even we're not planning this experiment. It was done in other animals. After someone said, have you tried, let's say, dogs, or have you tried this, or have you tried that? And when you did the other way around, it doesn't generalize. Like if you use parametric stimuli, it doesn't generalize to natural movies. But if you use natural movies, it generalizes to static images, gabors, dots, ping noise, and coherent random dots. The other thing that you can use this model is for decoding. And what we find in our data, at least in the visual cortex, this is, it looks low resolution because it is the mouse sees the images like low resolution. If you do this in a monkey, we're doing this in a monkey, you see basically what you see because the visual stimulus high resolution. But in mice, you see these sort of more blurred images, but you see there is correspondences between the image and this. Now, what is interesting in a previous paper, we found that if you want to build the best decoding model, if you build an encoding model first and you invert it, because you can invert it since you can backpropagate through the gradient, they're much more accurate. And we've done this with closed loop experiments versus if you build the decoding model first. And the reason we think that is because the loss function is much more well defined because you put the loss in the prediction of neural responses. When you build decoding, you're making hypotheses about what is the relevant things. Even in the motor system, if you're recording from an area that, let's say, that's kinematic, you put the loss in the dynamics, you may not get as good decoding. And that's what we find in the visual system. And I think this is an interesting thing that hasn't been extended yet in brain-machine interfaces and features. So far, basically, this model, this foundation model, we're putting it stimuli in behavior, and it's just reconstruct the stimuli and predicting activity, right? We're not putting the activity in. We're building new models that you put the activity in, like some of the previous ones that were shown. But if you think of this foundation model idea, at the end of the day, like all the inputs are the outputs, plus you have other stuff on the output that you can do, like you could diagnose a disease, or you could like predict, test the theory, or do decode it. But you can think of it like things that you measure on the one side, and you have like a gigantic neural network in the middle, and things you care about to study on the other. And in the way we've run it right now is like we built it so that it was stimuli behavior and mostly predicting activity, and use it as a digital twin to basically try and gain insights in what these neurons are doing. And that's what Catherine is going to talk to you about, some discoveries with this type of model. But another question is, can it do something very different in this model? And I think that's something that Blade introduced. Can it, for example, predict structure? Can it predict something that has nothing to do with neural activity and the network has never seen? Okay? Now, of course, it should at some point, because there is a relationship between function and structure. Like neurons that have different morphology, let's say, they should be doing something different. And to do this, we had this collaborative project with the Allen Institute and Princeton, where this is called the Microns project, and all this data are publicly available, where we did functional imaging of 75,000 excitatory neurons and did electron microscopy in a volume that contained 200,000 neurons, because it has astrocytes, neutrinos, all kinds of stuff, with 500 billion synapses. And this data is extremely valuable because in the same tissue, it's very unique. We have both structure and function. And, these data have been, there is a paper coming out that, the data is all publicly available, but there's a paper coming out describing the data, but it has data at different levels, all the way down to like, GAN-L levels, segmentation of nuclei and stuff like that, but also it has cell type information. And these data are also matched. We, especially Stelios Papadopoulos here did a heroic effort for 2 years to basically match the functional and the structural data together in alignment. So for the same neurons that we have function, we also have structure, okay? So in this paper here, what we did, and this was mostly work at the Allen Institute, they used morphological criteria and nuclear criteria for all the excitatory cells in this volume and classified them into these morphological and anatomical cell types, okay? And this goes from layer 2A down to layer 5 ET cells, and I think Blake showed some. And then we'll ask a very simple question. Can this model predict the cell types? The model of never seen morphology doesn't know anything about morphology, just has neurons. And it doesn't even know what area we're recording or what layer we're recording. We just like said, here is a neuron, here's the visual stimuli, there's this gigantic model on a different set of animals, not even the microns. But because it transfers very well to new animals, then we took the microns data and actually created a digital twin of this animal. Now you can download this digital twin and you can run experiments. Then any experiment you want, you can show you stimuli, you can compute stuff. And we can discuss if you're interested how you may want to use it. So this model is built in a way that its tuning function, or the way it predicts the response, is factorized to something that is very intriguing in neuroscience, the spatial component, I think where there is in a featural component. What is the tuning function of it? The classical thing is orientation tuning, but since I've shown you these neurons are not well predicted by double filters, this is this is an embedding in the feature space in this convolutional code. Okay, so you can think of each neuron having this feature component, the spatial component. And when you plot its spatial component, you get nice demarcation based on the retinotopy of all these extra stray areas. And you can read about this in this paper. Some archive is coming out soon. What is really interesting now, if you take the featural component and you cluster it using some unsupervised method, you can see that it predicts very well the visual area that is coming. So it's telling you we still do not know what exactly is different, and we can discuss it between these areas functionally. But we know that in this code, in this low-dimensional code, there is a difference that can predict very well what area between these four areas, what area it's coming. And it can also predict the cell type, the cortical layer that you're recording it, and also even within a layer, subtypes of, types of pyramidal cells. Let's say within layer 3, there is 2 major classes as classified by the analyst table here. For example, these are layer 3A and layer 3B. Now, the other thing that we wanted to use this model is to see if we can study something about the connectivity. Okay, and I'll try and go a little bit faster here. But what is interesting, let's say I'm a neuron and you guys are all possible postsynaptic targets. I send my answer something. Then I'm going to get close to some of you guys' dendrites, very close within 2-3 microns, and I'll stay close to it. But with some of you, I'll make connections, and with some, I will not. And we understand what is it that some neurons are selected by the neuron, the presynaptic neurons, to make connections, and others not. And there's this general concept of like-to-like connectivity. And here we did, we did 2 major things. One is that we looked at the signal correlations, and we looked at the axonal scale. That means that the co-traveling of an axon close to a dendrite, how long do they stay together? And the longer they stay together, the more similar their properties are. And also, if they do form synapses, the ones that do form synapses, the more, the number of synapses, basically, here, this is this one, the synapse density is correlated with their signal correlations. But then we wanted to ask the question, what part of the signal correlation determines this connectivity? Because neurons can be similar because they have overlapping receptive fields, or they can be similar because they have the same features. For example, just to make it simple, imagine 2 neurons that are both like red, but they have non-overlapping receptive fields, or 2 neurons that like red and green, and they have overlapping receptive fields. What dominates the probability that it will actually form connection? And what we find when you take this co-traveling into account, it's the feature selectivity and not the spatial selectivity that determines. So if I'm the neuron here and I'm red, and I go through a patch of cortex, I will be more sensitive to, if anything, there's a bit of an anti-correlation in some areas. And I will, if anything, connect with neurons that are not as much overlapping in the spatial domain, but they are red in the active. And this was only possible because we used this digital twin and we could model it very well, and we could factorize the tuning function like this. If you do this with orientation selectivity, it doesn't work, because as I told you, orientation selectivity, it doesn't capture this property. Okay? Yeah, I will skip this, but we've shown that this type of effects of like-to-like connectivity, if you just build artificial neural networks and train them on simple tasks, you get a similar effect. And to finish, back to this picture and back to the pictures that were shown before, which in fact, I think this will be together with Blake at some point, is that you can think of all kinds of data that you collect and you put them into this gigantic architecture and you try and learn this relationship that is essentially a predictive model, one way or another, whether it's forecasting or it's predicting one modality to another or within modality. At the end of the day, the way I see these things is that you're trying to make predictions. And ultimately, I think what we're aiming for is essentially trying to find an isomorphism or some sort of relationship, other than using it for treating diseases or other stuff that I haven't talked about and others may talk. We want to understand if there's correspondences or something that we can interpret and gain biological insights. And I've shown you some biological insights we're starting to understand by relating structure and function, for example, this connectivity rule would have been very difficult to discover if we didn't have this predictive modeling with factorizing between functions like that. And then I would like to, thank the many collaborators who have worked on this project and are funding, and especially Catherine Franke, who will talk more about what usefulness these models are. Thank you.